Hello friends, welcome to this video session. In this video session, I'm going to deal about a very basic concept, that is DNA packaging. So before diving into this concept, I request you to go through the basic videos that I dealt before, which mainly contains about the types of DNA and what is actually DNA, what is DNA made up of, the three major components such as sugar, phosphate group, and the bonds that are involved. So if you go through all those things, when you are clear with those concepts, this becomes very easy and simple. So what we are going to study here is how is DNA packaged, where actually DNA is present, what are the types of DNA. So it is very short video and all basics about the DNA packaging, etc. So we all know DNA is a genetic material. It is present in all the living organism and living cell. So DNA may be either single stranded or it may be double stranded. It varies with living organism. In humans it is double stranded, in plants it is double stranded, in certain viruses they are single stranded. And usually the DNA is a negatively charged reason. And and DNA can be packaged. DNA is very long. As I said to you, the DNA measures so many kilometers. It can cover the entire Earth 6 times, or it can reach from Earth to Sun so many number of times. Such long amount of DNA and contains huge genomic data. It cannot exist in the form of such long, because the cell is very minute. Inside the cell, inside the nucleus, is DNA is present. So this DNA has to be packaged. So that is what we're going to study over here. In prokaryotes, we all know that the nucleus is not well developed, the nuclear membrane is not very well developed. So the DNA is a form of a naked DNA. We call that as a naked DNA. So the DNA does not undergo any much complexity and all. It is very simple. It is just loosely coiled like this, either single stranded or double stranded, and it is associated with some protein substances here and there. It is associated with some protein substances here and there, and this structure is called as nucleoid. This substance is called as nucleoid. So this can be asked for any 2 markers if writing any entrance examination or other university examination. So what is nucleoid? Nucleoid is a naked DNA in case of prokaryotes, which are loosely bound and coiled, and they are associated with some amount of proteins, forming a complex called as nucleoid. It is very simple when compared to eukaryotes. When we go to eukaryotes, how is the packaging done? So we all know that they are double-stranded helical structure. Now these are negatively charged. These are negatively charged. They should be associated with something positively charged called as histone protein. So these histones are proteins that are positively charged. 8 histone units join together to form an octamer. Octa means 8. So 8 histone units. 4 histone units here, and the other 4 units will be backside, 3D. So imagine 3D. So 4 units will be here and 4 units will be here. So I'll just show you like this. So now this forms one octamer. There are 8 histone proteins, monomeric proteins, that combine together to form a 8 subunit called as an octamer. Now there are so many thousands of octamer that are attached. This DNA coils over this octamer. So if this is an octamer, this is a double-stranded DNA, it wraps around it or it coils around it in this way. It wraps around it, coils around it in this way. So this is one unit. So when you observe this electron radiograph, so when you see under electron microscope, it just looks like a bead on a string arrangement. So let us consider this as a string, and how the beads are arranged on it. How the beads are arranged on it. Bead on a string arrangement. The same way, this is the DNA strand, and this red color thing, the bead like thing, is called as a octamer unit. This is called as nucleosome. It is the unit, structural unit of DNA, we call it as. It is called as nucleosome. What is nucleosome? So the 8 protein which is coiled by the DNA, this unit is called as nucleosome. Like that, there are so many units joined that appears like a bead on a string arrangement under the electron radio microscope. So this is called as nucleosome. Now this is called as chromatin fiber or chromatin lemma. Chromonemma, it is also called as. Nemma means thread-like fibers. Now what happens? These like this, it goes on for so long distance, it goes on like this. This condenses, thickens, shortens, coils to form chromosome. So such long thread or filament called chromonemma along with this nucleosome. It thickens, shortens, reduces and coils to form chromosome. So this chromosome, for this process to occur, for this association to occur, you require a special type of protein called as NHC protein or non-histone chromosomal protein. These are not histones, non-histone chromosomal, because these proteins help in making this chromonema or thread-like filament into chromosome. This is done at the metaphase of the cell division or mitosis. It is done at the metaphase. So now what happens is all the filamentous structure like this gets coiled up to form a single chromosome like this, thickens and coils to form a single chromosome. This is the centromere. The center of the chromosome is centromere. This is one arm and this is one arm, and the tip of the chromosome, this is the tip of the chromosome, that is called as telomere. Telomere, centromere is the constriction, and this is the arm called as chromatids. So this is the, so this entire thread-like filaments that I showed you, it condenses, coils with the help of non-histone chromosomal proteins at the metaphase stage to form chromosome. And like this, there are 23 pairs of chromosome in case of human beings. There are 16, 5, 4. So it varies again with each and every living organism. Now, one more important thing about this chromosome, there are two regions you can distinguish in this chromosome. So that region of this chromosome which stains lightly. So you have a special stain to stain this chromosome, acetocarmine, acetoarsine, such these are the special stain that will stain only the chromosome. It won't stain other organs like chloroplast, ribosomes, mitochondria. It won't stain such. Only the chromosomes will be stained. So when you stain with such special stain, there are certain regions that take lighter stain and they are transcriptionally active. Such regions are called as euchromatin. Such regions are called as euchromatin. U in the sense true. They are the true chromatin or true DNA. They are transcriptionally active. Transcription in the sense it is a process where the mRNA is synthesized and they are loosely packed. Since they are loosely packed in those regions, the DNA is exposed and the RNA can be formed very easily. So they are transcriptionally active, they take lighter strain and they are loosely packed. These are the characters of euchromatin. And this part of euchromatin replicates first. They are done in the early S phase stage. Early S phase. You can see my replication video also. There I have mentioned this S phase. S phase is a synthetic phase of the cell cycle where the replication occurs. During the early S phase, this euchromatin region gets replicated. And simultaneously, there are other regions of the chromosome which stains darkly, which takes darker stain, and they are transcriptionally inactive. They are tightly bound, so they cannot be freely, they cannot be exposed, and DNA cannot be synthesized RNA such so easily. So they are tightly bound, they take a darker stain. Since they are tightly bound, they take a darker stain or they stain darkly and they are transcriptionally inactive. Such region is called as heterochromatin. Such region is called as heterochromatin. And these replicate during the late S phase stage. Late S phase stage. So this is all about the DNA packing. So DNA is not only present in the nucleus, we have non-nuclear DNA also. DNA is present in the mitochondria, it is also present in the chloroplast, and it is also present in case of yolk, or yolk in case of your sea urchin and all, it is present, and in case of your amphibia, frog and all, the yolk, ooplasm also, this DNA, certain amount of DNA is present. We call it as extranuclear DNA. Extra means other than the nucleus, where else the DNA is present, that is called as extranuclear DNA. So usually the DNA is present in the nucleus. Apart from this, we have mitochondria. They can produce their own proteins also, right? So that is the region mitochondria, and it is also present in the chloroplast. It is also present in the chloroplast. And in case of sea urchin, they say that it is present in the egg ooplasm. And in case of ciliates, it is present in the other regions also. We call it as and we call those as extranuclear. We can see it in case of the ooplasm of the sea urchin, reptiles, ciliate, paramecium. In such regions we can see. So this is all about the DNA, which is very basic stuff, which is very much necessary. These are like important things that you have to know. So I hope I made this video very clear, simple. Thank you for watching. Please do like and subscribe. Thank you.